Hey guys, it's Mr. Wolfe here, and I'm going to be going over targets 3 and 4 for genetic inheritance. And so, before we dive into everything, let's just look at targets 3 and 4. And so you might be looking at these and saying, all right, wow, there's a lot of vocab, and there is. And it's important to understand the relationship between these terms. So I highlighted them in different colors, especially for target 3. I want you guys to understand this relationship between what is genotype and what's phenotype. And then for these similar sounding words, homozygous and heterozygous, how are those different? And then, what's a dominant trait versus a recessive trait? So that's target 3, and it's vocab intensive, but it's important terminology to understand. For target 4, that's going to be I could explain Mendel's 3 laws of inheritance. And we're going to be focusing on the last two, the law of segregation and the law of independent assortment. And if you notice in this target, there's the word meiosis. And remember, meiosis is taking that diploid cell and making a haploid cell, our gamete cells right here. Because of how meiosis works and how chromosomes and traits are passed down, we're going to get these different laws. So the law of segregation and the law of independent assortment. So let's start where it started, with Gregory Mendel, the father of genetics. So this is the scientist that really provided the foundation for what genetics is. So this was a 19th century monk/scientist that did a lot of experiments with pea plants. And what I think is pretty amazing about this guy is that he knew nothing about DNA at the time. He just ran all these experiments with pea plants, and from his evidence was able to create this theory of how traits were passed on, his theory of genetics. And so at the time, there was this competing theory that traits were just blended together, that you might get a trait from the mom and the dad, and what the offspring would possess for that same trait would just be a blend between the two. But there was a flaw with this theory, and that is that eventually everyone would just end up looking the same. And we know that this is not true. And so this is not the way traits are passed down, and Gregory showed that with his evidence. So Gregory Mendel worked with pea plants, and he might have taken a purple plant and a white plant, or a pea plant with purple flowers, a pea plant with white flowers, mixed them, and he would have gotten, let's say, a purple flower. And then you could do this with plants. He could have self-pollinated this flower, so it's basically a purple flower mating with the same parents as before, mating with another purple flower with the same parents as before, and you get a purple flower, a purple flower, a purple flower, and then you get a white flower. And you don't always get this, but this could have been an example, and Mendel was confused or puzzled by these results. In his second generation, we had a purple flower, but even though it expressed that purple trait, it still seemed to hold on to that white trait and was also able to pass it down to the next generation. So this is going to be an example of skipping a generation, if you've heard the term before, and I know I got some questions in class about it. Right? And so one thing I want to go into is that explaining why this is that there are 2 alleles for one trait. So for instance, we might have a gene for flower color. An allele is just going to be the version of a gene. So the gene might be flower color, but the allele might be purple, or it might be white. All right? And another thing to keep in mind is that we have, as humans or plants or really any diploid organism, we have 2 versions of each chromosome, and that's why we're going to have 2 alleles for one trait. So on this part of the chromosome might be the gene for flower color, and we could have 2 alleles for that trait. In this case, you might get the purple allele from the mom and the white allele from the dad. Right? And so this leads into the law of segregation, which is going to be part of target 4. So what this states is that alleles are separated during the formation of gametes. And so here's the process of meiosis. We start with this diploid cell, and in the end we get our gamete cells that just have one version of each chromosome. And because meiosis works this way, maybe this chromosome right here might have an allele for the purple colored flower, and this one as a lowercase p is going to be the allele for the white colored flower. When you pass on your traits to the next generation, even though you have two versions of each chromosome, you could only pass down one allele, either this one or this one, to the next generation, but not both. And so which one gets passed down? Well, that's just going to be a 50-50 chance. Right? And so the fact that we have this law of segregation will lead into the law of independent assortment. All right. So this is showing meiosis again, and for the law of segregation, just going back to it, we have two alleles for each trait, but you could only pass down one of these alleles to the next generation. Now for the law of independent assortment, it states that genes on different chromosomes separate independently. And so during meiosis, chromosomes are going to line up, and this is where this law of independent assortment is going to come into play. And let's say for our purposes that these blue chromosomes are from the plant's, let's say the plant's dad, and the red ones are going to be from the plant's mom. Now, the dad chromosomes are not always going to line up on the left side, and the mom chromosomes are not going to always line up on the right side. They're going to line up independent of one another. And so you might have a trait on this chromosome for, let's say, color, flower color, and then on this one, let's say it's a trait for something about the roots of the plant. All right? And because they're on different chromosomes, they're going to separate independently. What do I mean by that? Well, they're going to line up just randomly. And it's going to be a 50-50 chance in terms of how they line up. And so here, just by a 1 in 2 chance, here the dad chromosome lines up on this side, the mom chromosomes line up on this side. And it's going to be completely independent, no influence on this trait, on how they line up. It's going to be a 50-50 chance on how they line up. And in this case, the mom chromosome just happens to line up on this side. The dad chromosome just happens to line up on this side. Now, this is just a simplified version. Keep in mind, for humans, we have 46 chromosomes. So there's going to be 23 chromosomes lining up here. And the variation we could get from them independently assorting is going to be quite a bit in terms of there's the different types of traits that an organism could get from its parents. Now there are times where independent assortment doesn't always come into play, and that's when maybe traits are going to be on the same chromosome. So keep in mind, law of independent chromosome is talking about different chromosomes. And so, sorry about that. So let's say, for instance, and we see this just from our observations, that the traits of red hair and freckles, we often see these traits together, and that's because both of these are going to be on the same chromosome and very close to each other. Right? And so since they're on the same chromosome, even when that chromosome splits up, they're going to find themselves usually together on that same gamete cell. And so that's when independent assortment won't work, is when those traits are on the same chromosome. All right. So just a quick review. You have to what for the earlobe gene, or really any gene? I don't know why I chose the earlobe gene here, but you have 2 alleles for any gene. And then what are the chances that you pass on the earlobe gene onto your kids, or in any allele really is what I'd rather put there. And that's going to be just 1/2, right? And that's going back to that law of segregation. All right, so let's look at a sperm cell, and it's going to be combining with an egg cell, 2 gamete cells making a zygote, now a diploid cell. How do we know if the offspring will have purple or white flowers? Because say right here is going to be the purple allele, here's the white allele. What's that organism going to look like? Well, before we go into that, let's just first identify that some genes or some alleles we're going to say are dominant, or some are going to be recessive. So I'm just going to skip this and go to this slide right here. All right, so I actually decided to go to the previous slide, so sorry about that. So what will, even though an organism might have a certain set of alleles, what's it going to look like? Well, first we're going to want to look at the genotype, and that's the set of alleles the organism possesses. And so for here we're going to represent the purple colored flower being a big P and white being a little p. So if we have one big P, one other big P, this is only going to give us a purple flower. And that trait of being purple, that physical representation of the trait, that's going to be our phenotype. So the genotype will lead to our phenotype. Let's look at our next, or our example all to the way to the right. And sorry that the key went away. So just really quick, big P is purple, little p is white. All right, so let's look to the one all the way on the right. We have two white alleles. What we're going to get is, and it's two white alleles for our genotype. What we're going to get for our phenotype is the white flower. Now let's look in the middle where we have one allele is purple, the other one is white. Well, in this case, we're going to look at one allele is always going to be, well, not always, but one allele might be dominant and the other one might be recessive. And we usually represent dominant being just more uppercase. And so what dominant means is it's just going to be expressed whenever we have these two alleles together. The dominant gene is going to be expressed over the recessive gene. So even though we have the gene for a white petal, because the purple gene is dominant, for our phenotype, we're going to see a purple flower. All right, so let's look at a new slide. Here we're going to be looking at the genotype, so just the alleles that have been given, so everything to the right of this line. Again, that dominant trait is going to be expressed over that recessive trait. So if we have two dominant genes right here, two dominant genes, it's going to be called a homozygous dominant. And if we look at the wording, the word homozygous, even though it might be a new word, zygous sounds like zygote, and homo is just going to be a word for similar. So it's going to be a zygote with similar alleles, in this case, dominant alleles. Right, so homozygous dominant is when we're going to have two dominant genes. And then we could say the same thing for the little p, the two recessive traits, a zygote with similar alleles, and these alleles are recessive, so homozygous recessive. And then finally, if we have our alleles are going to be different, big P, the dominant and the recessive, it's going to be called heterozygous, a zygote with different alleles. So that's all that means. All right, so that's our genotype right here, everything east or to the right of this line. And then remember that genotype, based on the pair of alleles that we have, it's going to give us our phenotype. What we actually see, or what we get, what's expressed is our phenotype.